This is the extra credit for the Unit 2 test. The first part of the question says to identify 3 macromolecules that are components of the plasma membrane in a eukaryotic cell and discuss the structure and the function of each. The plasma membrane is first made up of a lipid bilayer. And the lipids in this bilayer are phospholipids, and they're amphipathic, which means that they have hydrophobic tails, and these tails make up the inside of the membrane, and they have hydrophilic heads that make the outside of the membrane. And hydrophobic means it repels water, and hydrophilic means that it's attracted to water. And this structure gives the gives the membrane a semi-permeable quality, which means that only certain things can go through it. And ions or large polar molecules, so they have a harder time getting into the membrane without the help of proteins. And there are many different types of proteins in the plasma membrane. Integral proteins are embedded in the membrane, while peripheral proteins are not. And integral proteins usually form a channel which allows the ions and large polar molecules we talked about earlier enter the cell. And peripheral proteins have also other functions. They usually reside in the outside of the membrane, and their properties have to do with interactions between the cell with other cell proteins, and such as they regulate cell signaling and recognition, and they also take part in enzyme catalyzing. And another important part of the cell membrane that contributes to the fluidity of the membrane is cholesterol. And cholesterol is embedded in the lipid bilayer, and it plays an important part in maintaining the distance between each lipid. And this distance maintenance is also an important temperature buffer. And so when the temperature gets cold, the membrane wants to be more stiff, but the cholesterol makes the membrane more fluid. And the opposite happens in hot temperatures, and cholesterol makes the membrane more stiff rather than more fluid. The second part of the question says to explain how membranes participate in 3 biological processes. And as we explained before, cells must communicate with each other, and they release chemical signals by exocytosis, which is when the cell directs the contents of its secretory vesicles out of the cell membrane and into the extracellular space, which is just basically space outside the cell. And the receptors in the membrane allow signals to pass through the membrane into the cell, where the cell signaling begins. And I had some difficulty with cell signaling on the test, especially the G protein signaling, so I'll focus more on that process. And during a G protein linked reception, there is a protein receptor in the plasma membrane, like we talked about before. And there's also an inactive G protein and an enzyme involved. And what first happens is that a signaling molecule from the outside binds the receptor, which activates it. And then when that happens, the G protein is called over and it gains a phosphate molecule. And it goes from, the energy goes from GDP to GTP instead, which is the 2 phosphates to 3 phosphates instead. And that gives it energy, so the G protein can transport the GTP to the enzyme and activate it. And then the enzyme will produce whatever cellular response it does. And when that is done, the G protein loses its phosphate and it goes back into its state of rest until that happens again. And the cellular membrane also has an important part during the chemoosmotic production of ATP. And the electron transport chain has pumps. It pumps H plus ions or protons across the membrane, and this creates a concentration gradient throughout the membrane, and this allows proteins to move through ATP synthase embedded in the membrane to produce the ATP. And the third biological process I'm going to explain is muscle contraction. And first, the axon terminal releases acetylcholine, which binds to the other cell receptors. And then a sodium ion moves through the membrane channels, which depolarizes it. And this depolarization makes the Ca+, or the calmodium-dependent protein kinase 2, to get released from the smooth endoplasmic reticulum. And that's my video.